Okay, so I think we can start. So good afternoon and thank you for joining us. Welcome everyone to the February MCHRI Seminar Series talk. For this session, we have a really exciting talk. We're focusing on the Iterative 22q11 neuropsychiatry research program that we have here at MCHRI. And we'll be hearing from one of our new awardees that the program supports, Dr. Stanley and his graduate student, Michael Chavez, on their project, High-throughput neuroscience research and 22q11 deletion syndrome by a novel CRISPR toolbox. But first, I wanted to do a few housekeeping issues and also introduce the 22q11 program to everyone who's joined today. Just trying to move my slides. Sorry, hold on. Oh, okay, I'm going backwards. Sorry. Okay, so here we go. So this is MCHRI's education committee, and we wanted to thank them for all the work they do in putting on the seminar series and the annual MCHRI symposium. We've got a really amazing lineup of talks and speakers for 2022. And the next seminar in March will be from Dr. Rosen and Dr. Ojo on immune epithelial interaction in pediatric IBD. Please also save the date for October 20th for our 5th annual MCHRI symposium, which we are planning to hold in person. And tomorrow, our clinical research support office is holding a training session on EPIC. And so now I just wanted to give a brief overview of the Hamilton 22q11 neuropsychiatry program at MCHRI, which is supporting the work that's being presented today. The program aims to support research to advance our understanding and improve the neurocognitive and behavioral symptoms of 22q11 deletion syndrome. And it does this through supporting collaborative multidisciplinary seed grants and aims to bring together a diverse community of researchers at Stanford across many schools and departments who all have a shared focus on 22q11 deletion syndrome. And as a part of that, we've been holding, we will be holding a series of research seminars throughout 2022. And this is following on from the biennial symposium that we held in November jointly with the CDCM. So our first seminar is on April 7th, which will have a focus on immunology in 22q11. And most importantly, we'd like to extend our deep gratitude to Candace Hamilton and the Iterative Hamilton family. Their longstanding and generous support for 22q research at Stanford has really accelerated research and clinical care in the field. The neuropsychiatry program is really possible because of Candace's vision, passion, and commitment to supporting the highest quality research in 22q11 deletion syndrome that has the potential to impact patients and their families. And so now I will pass on to Dr. Katja Weinacht, our faculty director for the program, who will be introducing our speakers today.
Hello, everybody.
Thanks, Katja.
Hello, thank you so much for joining. I have the privilege and great pleasure to introduce today's speakers, who are two of my all-time favorite colleagues, Dr. Stanley and Michael Chavez. So Stanley does not need any introduction, but I'll introduce him regardless. He's an associate professor in the Department of Bioengineering associated with ChemH. What I didn't know until I studied his CV, he held a physics degree before embarking on his PhD in bioengineering at Berkeley, where he studied synthetic biology with Adam Arkin and, of course, CRISPR gene editing with Jennifer Doudna. And when he came to Stanford, or when Stanford snatched him in 2014, he took CRISPR editing a step further and applied it to the modulation of in vivo cells and of transcription. So two of the many claims to fame of Stanley's are CRISPR activation and CRISPR interference technology to switch on and switch off genes. And from there, he developed it further to epigenomic editing, 3D imaging, and many, many more. I skip over, Stanley, your infinite list of prizes and awards you received. Instead, I'm turning it on to one of the wonderful graduate students in your laboratory who I personally had a pleasure of working with, Michael Chavez. Michael is really a Stanford pup. He did his Master of Science here and then joined Stanley's laboratory to really use the CRISPR toolbox to apply it to fundamental problems of biology as difficult as making regulatory T cells, and then most recently to studying one of my favorite diseases is the wrong word, one of the diseases that is very near and dear to my heart, 22q11 deletion syndrome. And what is so fascinating and so difficult about 22q11 deletion syndrome is that it's a disease with incredible variability in penetrance and expressivity, and why it is that individuals that have the exact same gene defect have such different phenotypic presentations. We have no idea, but I think we'll know a little bit more after your talk today. So without further ado, Stanley and Michael, thank you for presenting today.
Thank you so much, Kadia, for the very nice introduction. Let me share my slide. If you can see the slides. Yeah, it's a great pleasure for Michael and me to be here to present the work that is made only possible under the support of MCHRI. And today I'm going to talk about the first part of the talk, give you an overview of the technology that we've been trying to make a better genomic research in neuroscience. And after that, I'll give to Michael, and he will continue to present about applying and making technologies customized to the 22q11 deletion syndrome. So the general interest for us is to make a better technology toolbox that allow us to better engineer the genome. And when we look at the human genomics, we were just amazed by how deep, how complicated that this genome confers. It has many layers of the regulation and the information. And it's our core belief that to make a much stronger technology that allow us to control every single aspect out of this genome, we need to control, for example, gene editing, the epigenetics, the transcription, and on a higher level, the 3D genome. I always think like a genome is almost like a newborn baby that we need to communicate with this genome, and being able to understand what they do, that what really people have been using sequencing to study or imaging. And on the other side, we need to influence this genome so we can change its output for the good, or correct the bad. So when we develop technologies, we have two particular objectives bear in mind. One is we would allow to significantly expand our capabilities to engineer and study the genome function. And I like to highlight function is a keyword here. Second is we will allow to enable robust and better in vivo research for both studying the function and the gene therapy. And needless to say too much about genome editing, which has caught eyes on many researchers and applying them to study biology in the past decade. In my mind, the unprecedented breakthrough come from CRISPR is high precision, because this molecule knows how to use RNA to target DNA in a highly programmable and highly precise manner. It has enabled a lot of super exciting advances for both research and for therapeutics based on, for example, gene editing to knock in genes, or base editing to focus on single base pairs changes. One key message that I like to bring up here is when we think about function, and it's more than the sequence ATCG. And when we think about genome editing, it's also more than editing. Engineering is more than editing. So that leads us to think about another platform technology that allow us to implement this type of concept to target anywhere we can and modulate the function come out from the genome, which is a concept of the nucleus-dead Cas9, Cas-D. It's an RNA-guided DNA targeting system. In particular, in our early work and also others' work that develops this nucleus-deficient DCAS by introducing mutations into this natural bacterial protein. And by silencing its cutting ability, this molecule become a targeting molecule. The key message here is this molecule can be used to target any gene, any sequence in any organism using customized RNA. So another analogy is we try to really just broaden these scissors by using a broken scissor. We try to broaden it to a type of like a Swiss Army knife like a tool. So that's why we call it a toolbox. I'm going to use a slide that is a little more too crowded to explain some aspects that we try to develop. Just give you an overview of what we've been trying to do without getting too much into the technical details. So in the past years, we've been trying to make a full toolbox consisting of multiple species of the CRISPR. In addition to the commonly used Cas9, we also been working on Cas12 or the RNA type targeting system called Cas13 and recently CasMINI. And all of them take advantage of using highly customized guide RNAs, showing this cartoon that you only need to define these colored regions, which is usually 20 to 25 base pairs, to base complementary to the gene of your interest, and figure out a way of co-deliver them into the cell, in this case, for example, a neuron cell, and either ex vivo or hopefully ideally in vivo, although I should say that in vivo editing or regulation still has been a challenge. But based on the nature of the molecules, we can exert DNA targeting or RNA targeting. This further leads to a full toolbox for genome engineering. And in one example, in our early studies, we use them for transcription control. This technology called CRISPR-I or CRISPR-A stands for inhibition or activation. The point is to fuse this nucleus dead molecule, for example, a Cas9 to a transcriptional repressor to silence the interaction between the promoter and the polymerase. So turn off the downstream gene very close to the target site, or fuse that to an activator like a tripartite VPR system to activate, and by recruiting RNA polymerase to the promoter to upregulate the nearby genes. And expanding the concept leads to another control on the epigenetic level, which is in addition to recruit the transcription effectors, we can also co-recruit the epigenetic enzymes, for example, the DNMT, DNA methyltransferase domains 3A and 3L, and combined with transcription control can rewrite the DNA methylation very close to the target site. And compared to the global epigenetic modulation, this is very precise. And the interesting observation is this also leads to silencing of the nearby gene, but in a permanent manner. So basically, by recruiting the DNA methylation can create a long-term silencing memory. And in a similar manner, can co-recruit the activator VPR and also P300, which is a core unit of the acetyltransferase, to rewrite the nearby histone H3 lysine 27 acetylation to establish a longer-term and a stronger gene activation memory. So further expanding the concept, it leads to another technology called CRISPR-Go, stands for genome organization. The question arises from, what if we reposition a piece of DNA inside the human nucleus from location A to location B? For example, location B can be nuclear membrane, location A can be interior. And what happens to this gene expression, epigenetics, and also long-term DNA like behavior like recombination? We did this, make a technology by recruiting the abscisic acid. It's a chemically inducible system to target any gene and of interest to, for example, nuclear membrane. And further, we use the technology, we make technology for chromatin looping. As you know, the interaction between promoter enhancer usually controls activity of the nearby genes. And in this technology, we recruit two clusters of Cas9 to two nearby locus separated by 200 kilobases. And using an also similar abscisic acid chemically inducible system, we recruit a heterochromatin protein 1, which is a scaffold protein to each cluster. The cluster will recruit many copies and grow in condensation manner and grow in size, and ultimately they merge into a single locus. And using that method, we create a synthetic looping between distal DNA and study what happens after we create that looping. And recently, we've been expanding technology to study many enhancer functions. For example, some genes like oncogenes or neuronal like stimulation genes in many enhancers conditional to the stimulation. And some enhancer can be proximal, some enhancer can be distant, which is more than 1,000 kilobases away. We try to make technology to study either individual or pairwise enhancer function and study what come out by perturbing them. And finally, recently, we've been also using the technology to target RNA. In particular, fitting to the pandemic's need, we target the SARS-CoV-2 RNA genome and show that, like using Cas13 system, we can degrade the RNA. So this actually leads to our philosophy is we would love to expand the toolbox, and for every single application, we customize particular genome engineering tools that will be robust, efficient to study the problem. And the outline of the talk today is I'm going to give you two particular examples, both partially supported by MCHRI, on how we further make technology useful for in vivo research and with a goal for the next stage of neuroscience studies. And then I'll give the stage to Michael to talk about how we can apply them to 22q11 deletion syndrome. So as my first example is how we can enable CRISPR for in vivo use and applications. I want to mention one big barrier in the whole genome engineering field, even using CRISPR, is the molecule is very large, and its very large size of this molecule dramatically stops us from many super exciting applications in vivo. Just take the commonly used Cas9, for example. It has a large size, about 4.2 kb DNA encoding this single molecule. Although it's very efficient, but now the question is, how can we first deliver it into the cells or into the body, right? How that efficiency can be satisfying for applications. Example is using adeno-associated virus, AAV, and which has limited packaging capacity, 4.7 kb, and the CRISPR itself takes about 4.2 kb, making it extremely hard or sometimes impossible to deliver to many type of cells. How to solve this problem? So we've been thinking how to make a smaller gene editing molecule. And luckily, and there were research done 2-3 years ago by Jennifer Doudna's lab on reporting another type of CRISPR called Cas14. It's a different subtype of CRISPR. And what's intriguing about it is it has only a 1/3 size of Cas9. So rather than 4.2 kb, it's only 1.55 kb. You know, it's slightly larger than GIP, which is super nice for thinking about its broader applications. And what makes it possible to use such a small protein, yet still achieving the RNA-guided DNA targeting is, unlike Cas9, which is a monomer that binds to guide RNA DNA, Cas14 is a dimer that binds to a single guide and binds to a piece of DNA. So that could explain why it still works at a small size molecule. However, to our disappointment, when we first tried to use this Cas14 by fusing to an activator to target a gene in human cells, we found it doesn't work at all. Actually, while disappointing, it was not surprising at all because most CRISPR systems came from bacteria, and the bacteria host has genomes 1,000 times smaller than the human genome. A system has been efficient in a bacterial host doesn't mean that it can work in human cells due to the 1,000 times larger search space for the molecule to find its target. So following this argument, we think, okay, is there some way that we can do engineering to make the system 1,000 times more efficient? Then we should get a system that works. And that's what we try to do. I'm skipping a lot of engineering step in the middle, but the concept is a molecule, like a Cas molecule, like Cas14, it doesn't work well in human. It's because it doesn't have good binding and interaction with DNA. And how to make this binding better? We should make it more positive charged. The DNA is negative charged, and if you make it more positive charged, it likely can search the DNA binds faster and binding tighter. That's why we selected many residues and mutate that to arginine. Showing some data here, we did some systematic structure-guided design to mutate individual residues to arginine to increase its surface charge. And so interestingly, even some single mutation can enhance the activity, like by more than 100-fold, measured by like a GIP reporter expression from the HEC293 cells. And that really gave us some confidence saying, okay, now let's do more engineering into this naturally very mediocre, very bad system. And then we did multiple rounds of protein engineering evolution. Each round, we introduced one more mutation based on the previous round's successful mutations. And after 4 rounds, we were able to get some mutants bearing 4 mutations distributed around the protein, which almost showing a 1,000-fold improvement over the wild type. And how well this system works. Of course, let's compare that to Cas9 and also Cas12. And for Cas9, as you can see that we fuse this dCas9 or dCasMINI to different activators. And the gray bars show Cas9 and the blue bars show CasMINI. In terms of fold activation by 24 hours, CasMINI actually went, and which called so it actually shows that it's faster for activating a gene. And at 72 hours, they are comparable in many cases, and the CasMINI seems to work even better with some smaller activators. And we also compare that to a Cas12 system, and by testing on endogenous genes like interferon gamma and human hemoglobin, we find using a single guide or using 2 guides to direct each system, and we find they work comparably well, or sometimes CasMINI works even better. In addition to target gene regulation, we also found this CasMINI allows fairly robust gene knockout and base editing. For example, we were able to use this just nuclease version of the engineered CasMINI to cut genes at different locations. And we found that besides the decent amount of efficiency, which now we got like 20–30% cutting efficiency, the cutting window for CasMINI is actually wider, and also it's wider than Cas9. This makes it a better system for gene knockout. Cas9, in comparison, only disrupt 1 or 2 base pairs. CasMINI can disrupt a window like 30–40 base pairs. That's why it's better for knockout. For base editing, for which converts a precise adenine to a guanine, and we also find it has some efficiencies there. So a summary for this part is this: we made a small gene editor molecule, which can be used individually or fuse that to different effectors. And the size is still very compact, make it suitable for delivery using AAV. And as a second example, I'd like to highlight another example, which is how we make a multiplex genome engineering in retinal neurons. And shown here is an image that's regulated by our engineered CRISPR. Again, we have a goal here, which is how can we control more than 1 gene, multiple genes in the genome, hopefully in vivo. And the reason we ask this question is if we look at a disease like 22q11 deletion syndrome, it's not affecting 1 gene, it's affecting many genes. And it still remains unclear if it's a single gene or it's a combination of multi-genes contributing to the problem. And if we were trying to solve this problem, should we control a single gene or should we control many genes? And I tend to think we need to control multiple genes as it may restore better function back. So the core concept of using Cas12 is a CRISPR system that can take a CRISPR array. You can easily just arrange these 50 base pair guides in an array, and activating guides and repressing guides, and you can just and the Cas12 will go there and to process each small guides out of the single transcript array, and then do some individual gene regulation work. And if we think about genes as a network, this is particularly true if there's like certain genes that we need to upregulate, certain genes we need to downregulate to restore a function of the program. And we would like to make it highly compact and ideally contained on a single vector, so we can easily inject to a brain or somewhere, right, eyes, so it can help its function on single cell level. And conceptually, and these different guides come out to either activate genes to mediate like different cell states, could be like a cell type or could be a function come out from neurons, and also simultaneously inhibit different functions that you don't desire. Okay, as an example, I'd like to show one early test that in collaboration with Sui Wang's lab in ophthalmology department and led by instructor Lucy Guo. and to test how we can use this multiplex genome engineering in retina neurons. And we try to activate 3 pluripotent genes, AUT4, SAS2, and KL4, directly in endogenous location into the retina neurons. And because previous reports show that this might promote the neuron growth and regeneration, give some benefits to this retina physiology. And that's why we encoded a single array, have 6 guides activating 3 genes. And we also encoded a Cas12 fused to a mini activator, injecting to the eyes, retina intravenously. And then after 21 days, we detect what's going on. We actually have 2 methods of delivery. One is using in vivo electroporation, and second is, I show later, using AAV. And if you dissect the biopsy of the neuron part, it's a multi-layer situation, and we try to image different layers, and by detecting what the delivery efficiency, you can see the GIP marker, and what's the efficiency for Cas12 expression, which is the HA tag, and finally, what is a target gene activation? Well, you actually don't see much gene activation here. There's not much co-localization between the Cas12 and the target gene, suggesting it's not very efficient. That's why it promotes us to further engineer this system by introducing mutations. And I'm going to skip this part for protein engineering, which is what we frequently do in the lab. And at the end, we got another mutant for we called hyper Cas12, stands for hyper efficient Cas12, which has 979-fold improvement on the efficiency over the wild type system. And we use this hyper Cas12 and showed how we can make that better genome engineering. In one test, we injected 1 wild type and 1 hyper Cas12 into 2 eyes, 1 eye each Cas12, and combined with the same guide RNA. To knock out a YP, which is just a proof of concept to quantify efficiency, then dissected the retina to see what's the efficiency for knockout. As you can see, comparing the wild type to hyper, and we tested 5 mice, we frequently see that hyper Cas12 gave much better knockout on the YP compared to the wild type. And since this is done pairwise, so we think this is actually pretty true for different genetic background. And second is we use this hyper Cas12 and to simultaneously activate SAS2, KL4, and AUT4. I'm showing 2 genes here. And so again, we did the same experiment as I showed in 2 slides earlier. And we detected Cas12 expression, and we also imaged co-activation from SAS2 and KL4, etc. As you can see in this case, we got very nice SAS2 and KL4 activation, which nicely co-localize with Cas12 expression, suggesting it's likely due to the CRISPR activation, induced activation. So finally, we also found that by activating these 3 pluripotent genes endogenous in situ to the retina neurons, we find a high percentage of migraine neurons moving from the inner nuclear layer into the ganglion cell layer. As you can see, these cells that only present in the hyper DCAS12A activation, but not in the DCAS12A activation because it was fairly inefficient to do so. And we also compared using the hyper Cas12 by activating 3 genes versus by activating just a single SAS2, AUT4, or KL4, and by quantifying the percentage of cells, Cas12 positive cells that migrate to the IPL and the GCL layer, we find activating 3 genes has about 25% migraine neurons in this situation. A summary about this part is we now have a technology that we can feel comfortable to control 1 to 20 genes. I mean, I'm saying 20 genes because sometimes it works for 10 genes, sometimes it works for 15 genes, but the number can be pretty large. It's definitely more than 2 or 3 genes. And combined on a single vector, we can control many genes in parallel. It's very efficient. It works for activation, repression, and editing. And also we look forward to test that for in vivo cell reprogramming, CRISPR screening, and consider some more polygenic cause of the genetic disease. With that, I'm going to give stage to Michael.
Great. Let me just share my slides real quick. Great. So how do we apply this now to 22q11, right? And to start off with, we just took a principled approach of looking at what people had done in the literature before, right? Let's not engineer for a potentially solved problem. And when we looked, we noticed that there was two types of literature out there. There was people who were doing what we call reverse genetics, right? So they were taking wild type cells and they were either using CRISPR or something similar to knock out things in a 22q11 0.2 locus and to see like how they became closer to 22q11 DS cells. There was also the forward way, which is you take 22q11 DS cells and through lentivirus or AAV, you introduce cDNA to see if they become more like the wild type cells. And when we were looking at the forward, which is the most exciting to us because we're interested in future of gene therapies, we noticed that people were doing it in a very similar way that potentially was creating not very good information for the field. And so when we look at this, we noticed that when people were introducing genes, and here I'm kind of showing a lentivirus type thing where you introduce it via genomic integration, everybody was kind of using this ability of synthetic promoters. So you would use an unnatural promoter to drive cDNA expression and hopefully rescue the phenotype. But from previous knowledge of things we had been working on, we knew that this might create as an issue. And the reason comes from dosage issues, right? So if we think of a wild type cell having an expression of 1 for one of the deleted genes, and we can think of 22q11 DS cells as having half of this expression, then using unnatural promoters often leads to a way overshoot of rescuing that phenotype, right? We get somewhere, in this kind of toy example of 3 units of expression. And in many cases, when you look at the literature, overexpressing these genes actually creates its own disease pathology in itself. And so while we had really interesting tools, we weren't really correcting gene dosage. And so we thought, what could we do with our technologies that could allow for precise gene dosage correction? And so I'm just going to go through how some of the things Stanley was talking about is now being applied in our lab towards this 22q11 disease. So to start, we're just going to start off with our CRISPR-A molecule, right? And so Stanley went deeply into this, but essentially the way this one works is all you have to do is create a guide RNA that targets just upstream of your gene of interest, so somewhere in the transcriptional start site. And we are looking at all the options that we have. So we can use traditional Cas9, but we also have opportunities to use the multiplexing of Cas12 or the kind of more translatability of CasMINI as we move forward here. But in general, all you need to do is create a screen of various places to bind this on the transcriptional start site, and through that are able to get some of the mRNA back that are missing in these disease cells. A huge issue with using CRISPR-A in these instances, though, is that it easily silences, right? So anytime you put it into a primary cell, CRISPR-A will be gone within 24 hours. And so we really had to find a way to engineer around that piece such that we could start looking at the neuron stage and having active CRISPR-A in those neurons. And so we spent last year kind of thinking about how we could potentially do this, and we tried a bunch of different options. But the one that worked really well was using a transposase system. So in this system, what we do is we co-deliver 2 plasmids, the transposase itself, and then also this plasmid that contains a CRISPR-A driven by a TRE3G promoter. And this TRE3G promoter only activates when you induce it with doxycycline. And the beauty of this is we can engineer cells and create clonal cells at the iPSC stage, differentiate them all the way through neurons, and then induce with CRISPR-A, allowing us to kind of create some of these screens at the neuron stage that wouldn't be possible without this technology. And just as a start, we showed that this actually works in iPSC. So if you see on the right here, we have two different cell lines, one on the left without the CRISPR-A circuit, and one on the right with the CRISPR-A circuit. And when you induce with doxycycline, we can activate the natural locus CXCR4. And then the second technology we're thinking about is a new technology that we call CLIP, which is CRISPR for long-fragment integration via virus-like particles for HDR, right? And so the way this one works is we were looking at the field also of knock-ins, right? And I'll talk about why we're excited about knock-ins in a second. But essentially, when you look at these, the field, most of it is done using either AAV vectors or some kind of PCR DNA or plasmid. The problem with this is packaging limit and toxicity, right? So AAVs are great and it shows really, really good data. But unfortunately, if you have a much bigger cDNA or want to deliver multiple things, you only have kind of, as Stanley was talking about, 4.7 kb to deliver. And many of the genes we're interested in here, or even doing doubles, as Stanley had talked about, are going to be bigger than this packaging limit. And so we needed to find a way to adapt other technologies to that allow us to create targeted knock-ins in genomic locuses. And so we create a clip, which is essentially, we create a virus-like particle that's based on lentivirus, but does not have the ability to integrate into the genome. And it delivers this payload, which is going to be an inserted gene that we want to deliver to the cells, followed by homology arms that allow it to kind of do HDR and integrate it directly into a spot on the genomic locus. And then we've added these cut sites right here that are super important, but I'll tell you why in a second. But essentially what happens is after we deliver this virus-like particle, we wait 24 hours in the cells and we introduce Cas9 RNP. This RNP not only targets the one of the genes that we're excited about, so in this case it would be a 22q11 gene on the undeleted locus. It also cuts away the LTRs of the gene, the lentivirus, the virus-like particle. We've changed the wording there every once in a while. But anyway, so it cuts it off and it allows it to create a much, much better HDR template that then induces HDR on one of the genes that we're excited about. And so before I go into how we're going to use this, just to show you kind of the ability that this clip method has, right? So essentially, we just started off by taking a GFP and trying to insert it into the actin B locus right upstream of the N-terminal methionine. And when we do that, we show that the cut sites are actually really essential to creating a high efficiency knock-in, right? We get an extra 20 to 30% boost in efficiency by just simply adding these genetic additions right there. When we move over to primary cells, where this is actually going to be used, we started off with primary T cells, and we showed that, you know, with all these additions, we get a really robust efficiency with high efficient introduction of GFP into the actin B locus, and it's across donors. And so we've created a way essentially to introduce knock-ins without a lot of the issues. And one of the biggest issues is toxicity, right? So when we think about things that could have a 9.6 kb payload size that could be delivered, we have to take AAV out of the equation, but there's two other options out there. You can deliver plasmid-based DNA as a DNA donor to cells, or you could directly create linear DNA by PCRing off of a donor. And so we had heard a lot about the toxicity that naked DNA creates in primary cells, and we just wanted to test how ours stood up to those other two options. And so again, we just delivered GFP to the actin B locus using both a plasmid and a PCR. And in this case, it was again primary T cells, but primary cells in general have this DNA toxicity. And as you can see on the left, the clip method is has very, very low toxicity, especially in comparison to the other two. And so what's important here is that allows us to create some of these clip knock-ins that would allow us to kind of move them again from an iPSC into a neuron without having to create a ton or baby these cells for a very long time, creating one single cell. And just to prove that this has a much bigger payload than maybe AAV could possibly do, we also delivered one of our hyper Cas12s, which is about 5.5 kb in size, and we were able to knock it directly into the actin B locus and show that we can get about 20% expression of this gene. The other great piece of this is because it has such a big payload limit, we can knock in two things at once, right? So we can encode two separate knock-ins on one clip donor, right? And so what you're seeing at the top here is a GFP being delivered to the actin B locus and a MCHRI being delivered to a RAB11A locus, which is just a housekeeping gene. And this is all encoded on one single clip donor. And when we deliver that to the cells, if we just deliver it with an actin B cutting enzyme, we get HDR in the actin locus. If we just deliver it similarly with the RAB11 guide, it creates high efficiency in the RAB11 locus. And then if we deliver both, we do get a population of cells that have now both genes with genetic knock-ins in them, which is a really exciting technology because this already by itself, even with just small, insertion of fluorescent proteins is already bigger than the packaging limit that can be seen by other opportunities. And so we get that beautiful low toxicity, high efficiency, and high payload using this method. And so how do we apply it to 22q11 and how are we doing this in 2022? So the most important piece of CRISPR-A, going back to the VPR piece, is we can direct these molecules to different parts of the transcriptional start sites upstream of a gene. And the beauty of doing this is by choosing the location, you actually get different amounts of upregulation of the gene you're targeting, right? And so what you're seeing here is 3 different guides targeting different pieces of the DGCR8 transcriptional start site. And in this case, we can get, anywhere between 1 and, 5x, for example, upregulation just by changing where it binds to. And the exciting piece about this then is when you have a disease like 22q11ds, we can essentially target about 100 guides per transcriptional start site. And when we do that, there's likely a very high likelihood that we'll find a 2x one that'll precisely correct the gene dosage. But we don't need to know anything about it beforehand. We can run a full screen and just treat each single cell as its own experiment, right? And so to do this, what we'll do is we'll create 100 guides per gene, so about 5,000 guides. We will transduce iPSC that have our CRISPR-A, doxycycline inducible CRISPR-A into it. We'll differentiate them to neurons, and then we'll add doxycycline. And what will happen is each single neuron will have upregulation of one of those genes that have been deleted, creating an upregulation off of the haplosufficient locus. Then we will use we'll use some kind of heuristic that allows us to say these cells have an improved phenotype over these cells. And through sequencing, we can say that this guide created this amount of upregulation of this gene, and therefore is a great candidate for further a dissection to see how it rescues this neuropsychiatric phenotype. When we have those in hand, we now have a short list of things that are essential to kind of helping create a better phenotype. And what we can do is now uses CLIP's ability to actually create that gene correction. And so the idea here is if we use CLIP to knock in a cDNA off of its endogenous locus, we can get 2 proteins off of 1 locus and all the good regulatory elements that come with it, right? So if we go back to the DGCR8 example, right, we can introduce a cDNA that creates that upregulation, and we get 2 proteins off of 1 locus, overcoming the haploinsufficiency that people suffering from this disease see. And so, we can just take a short list of these hits, introduce the knock-in using a high-throughput screen that we've done, and see exactly how, if both on a differentiation level and at the neuron stage level, if we can rescue some of the phenotypes. And so the way we're going to do this is using the hits. We will create 5 single knock-ins from our 5 favorite hits and 10 double knock-ins using CLIP's ability to do 2 off of 1 lentivirus particle. And we'll use bulk RNA sequencing and electrophysiology to see how these corrections, which have the potential to be a future gene therapy, could move forward some of these interesting targets. The only problem we're currently having, and we'd love to hear people's thoughts if anybody has any, is we really need help deciding on what to phenotype off of, right? So the screen requires that we have something that says these cells are now different or maybe improved over these cells. And it needs to be on the more high throughput side, right? So we can take things like transcriptomic signatures or protein biomarkers, or we could even use like genetically encoded calcium reporters that tell us like these cells have an improved phenotype. Things like patch clamp, unfortunately, are interesting, but don't allow us to do the high throughput screening that we would need. And so if anybody has any thoughts, we would love to hear them, especially in the Q&A, or please message us after. And with that, I can leave it back to Stanley.
Right, this is just, what I want to mention at the end is first, thank you to MCHRI for the support to make this project possible. And second is, I think there's a big difference between a technology that only a single lab can use versus a technology that every lab can use. So we try to make technology that can be broadly useful to the whole community. And if you find any of these capabilities on the genome side that relate to your research, so we'll be very happy to offer help. Thank you. And the next one is a acknowledgement slide, Michael. Yeah. Thank you so much for the funding, and of course our collaborators. We have really had this program has fantastic collaborators like Katia been teaching us on the IPS work and related to the disease, learned so much from her, and Marius and Sergio all provided lot of advice on the neuroscience side. And Sui, Yanggu, Yangsong from ophthalmology department provide help for get access to test the technology in vivo. And thank you.
Wonderful. So thank you so much for this fabulous talk, Stanley and Michael. Of course, you see my eyes start to light up when I see the entire CRISPR array and that you can switch so many genes on, because not only does this apply to 22q11 deletion syndrome and the neurons, but to any other tissue, for example, the thymus, that you want to make. But we bring it right back on target now, because I'm sure there are many people who have lots of questions, and I'm sure you'll be flooded with requests for collaborations after this talk. But I'm just going in order right now, and I invite everybody, post your questions in the chat or message or whatever feels appropriate, but I'll go in order of appearance. So Jeff Rubin is asking, what are the VLPs used in this study composed of?
I can take that one. The VLPs are based off of a lentiviral backbone, but they are integrase deficient. So we have done a couple mutations and a couple extra gene modifications that allow us to make them much safer. They don't integrate anymore.
Okay, great. Thank you. Another question comes from our own Casey Gifford. Hello, Casey. Thank you for joining. And Casey is asking, do you have plans to perform the rescue screen during differentiation towards other lineages that are also perturbed in 22q11? I'm sure she's asking about the cardiac organoids, for example.
Yeah, I can take this one too. So the beauty of it is that the screen is infinitely usable again, right? And the IPS are infinitely usable again. So they can really be applied. I think the main thing here is that we need, again, some kind of phenotype that is easily screenable, right? Something that allows us to, either through staining or through a transcriptome signature, something high throughput that allows us to say these cells have been rescued. And we don't currently have plans, but of course are super excited about the thymic niche, and also would love to hear anybody else's thoughts if they're excited to work with us.
And Michael, allow me one clarification question. So the IPS are stably transduced, and then you perform whatever differentiation protocol in essence you want, and then you switch on the docs at whatever stage you are at, and then anybody in the community who is interested in using this tool for whatever lineage they are interested in can use this, right?
And actually, it even goes one step farther, because I think the guide library can also be really irritating to clone and put together. But because they're all the same genetic deletion, by making the library, even that piece is available to people. So there's no actually extra engineering to apply it to other types, other phenotypes.
And then for the guide library, that is a complete mix and match and director's choice, right?
Yes.
Well, infinite opportunities. Great. So Jeff Rubin is asking another question. So thank you. One more question. What type of sequencing are you utilizing to ensure your clip integrations are on target?
Yeah, we are actively working on that right now. Actually, it's a timely question. We're hoping to employ long read sequencing to see how often we get off targets. I'm assuming there's going to be some, but the beauty of it is because we're knocking it into protein coding locuses, you can usually tell by flow cytometry, the difference between an off target, which is going to be a lot weaker in expression, and an on target. And so we're pretty comfortable that it's on target, but we are using hopefully long read and then potentially just NGS to read it out. Stanley, I don't know if you want to add there.
Yeah, so I think in the gene editing field, there have been a number of sequencing methods developed, including like guide-seq, and more recently there's like some one-seq. There are a lot of names there. Like the principle is trying to detect like hopefully unbiasedly. So we are going to apply some of technology like a guide-seq like technology to detect if off-target versus on-target. To establish that. That's also what we got for the manuscript revision.
Yeah, that's a
perfect. And then Tony is asking, Tony Oro, thank you for joining, is asking a question that has also been burning on my mind, giving the link between microRNAs and 22q11 deletion syndrome. So Tony's asking, can you discuss regulating expression of microRNAs given the DeGeorge critical region 8 possible connection?
Personally, I love to learn from like Bethany, like about the link between microRNA and the DGCR8, and I know DGCR8 probably is involved in the processing of microRNA. Are you asking like how that upregulating DGCR8 influence the processing, or do we directly control the microRNA downstream of the DGCR8?
Potentially both, right? My question would pertain to both. So you have haploinsufficiency of a gene that controls microRNAs, and then there is at least a few people who have implicated direct changes in microRNAs in 22q11 deletion syndrome. So could you either target the microRNAs, or can you just upregulate the George critical region 8 and screen for an effect? Tony, is that what you had in mind? Yes.
Yeah. Okay, so for our current research, we haven't done too much regulation directly on the microRNA. And a couple of years ago, when I did some study into the topic, we think like some microRNAs have many repeats in the genome. They processed from the pre-transcripts. So that's why, as of today, I'm not 100% sure how efficient that would be, and if we do it, where should we target? But that's a good, interesting topic for the next step. And regarding the like DGCR8 impacting the processing microRNA, I guess that's a interesting question that maybe we can done by perturbing the DGCR8 and measuring the microRNA as an output to perform sequencing and to see how dosage of DGCR8 impacts the properly processed.
Exactly. And DCGR8 is one of the genes in the haploinsufficient region. So you're already starting out with a 50% reduction. And then, you know, if you perturb it on top of that, exactly right. And so Tony has another question that was also burning on my mind. So Tony's asking, have you validated continued expression of CRISPR A and I targets after differentiating iPSCs to different lineages of neurons by single cell RNA sequencing, for example?
I can give one example, and Michael can chime in as well. So a couple of years ago, we did publish a work on using CRISPR-A mostly, but also some work from CRISPR-I, put that into the embryonic stem cells and differentiate that embryo stem cell into neurons. Here a 2 to 3 week differentiation protocol. And we did find that there were silencing, and mostly on the protein side, like Michael mentioned there. So the strategy that we used was to inducibly express this system, and also by creating pre-validated stable lines, how this CRISPR-I, CRISPR-A in the genome, and so make sure they don't silence much. And the interesting question I have today is, with the smaller size, for example, if you use CasMINI, which is a third size of the Cas9, and the overall regulator, CRISPR-A now used to be 5.5 kb, now get down to less than 2 kb. I'm curious to see, we haven't tested yet, like how much silencing we get from these much smaller proteins. And if you know the answer, we're allowed to share. And Michael, do you want to comment on the sequencing side?
No, that was what I was going to say.
Yeah, so the downstream, like a single cell RNA-seq, I think this is very interesting because there were some like work into the perturb-seq. Basically, by perturbing a gene or a pair of genes in the genome and perform single cell RNA sequencing to see what come out by this input. And to our knowledge, that hasn't been done so much on the differentiated iPS. That's why, like, what we are very excited about this project as well, like by creating a CRISPR-A library, perturbing these haploinsufficient genes up of 1 or 2 or 3 of them, and we allow to perform this sequencing and see how much on the transcriptome signature side. And we allow to test hopefully by measuring tens to hundreds of perturbations in a single reaction. That's our goal here.
And let me build on one of Tony's questions. So you said the CRISPR-A, CRISPR-I effect usually wears off after about 24 hours. Did I understand that correctly, Michael, when you presented earlier?
if you don't have an inducible system, like by the time you have lentivirus on them, they will silence, which is why we switched over to this particular system.
Okay, so then you said, you can take it one step further by using either methylation or acetylation to induce stable epigenetic changes. Have you used the epigenetic editing also in your work on recapitulating differentiation patterns of tissues to see if you can have a more permanent effect?
Currently, we haven't done that. The current epigenetic editor effector that I'm not going to hide that is they are pretty large. For example, the DNA methyltransferases, those for silencing, the size of the effector themselves adds up to 4 kb. And the phenyltransferase, which we can get much better longer-term activation compared to just using the trans activator, and also very large, 2.5 kb. So it's one of the topics in the whole field is how to minimize the compactness of these epigenome effectors. People have spent, including us, spend a lot of time on transcriptional side. Yeah. Now, like we're shifting more and more into the epigenome side. And,
fantastic. Very cool. With that, I'll invite the audience. Any other questions that might be coming up right now? I also see we are relatively close to time. So one more, I'm putting a plug in. If you have any other ideas, as Michael mentioned, what are the perfect tools for readout? Of course, transcriptional, always our readout, protein, always our readout. Michael, you said you have stably transduced approaches for calcium sensors.
Optogenetic approaches for calcium readouts too.
Okay, so if anything else, you know how to get a hold of Stanley and Michael. And with that, Rachel, do we have any other announcements for the next seminar?
I'm just going to put a link in the chat to everyone to invite all the attendees here to our next seminar on April 7th, which is Spotlight on Immunology, and we've got a great lineup of speakers for this event as well. Dr. Kathleen Sullivan from the Children's Hospital of Philadelphia, Dr. John Sleesman from Duke University, and Dr. Andrew Jennery from Newcastle University. So I do encourage everyone to who's interested to register for this as well as the next MCHRI seminars in March.
Perfect. So then I thank everybody for attending today. Thank you so much for the speakers. We're looking forward to many more collaborations and many more follow-up talks from this. And we'll see everybody soon. Bye. Thank you so much.
Thanks very much.
Bye. Thank you.